So good morning, everybody. It's lovely to be here today, and I'm under very, very strict instructions to keep to my time. So before I start my timer, I just would like to take this opportunity to first thank you all for this opportunity, not only to be here today, but to thank you for all of your contributions that you make to Garvan and to the kind of work that we're doing. So with that in mind, I will actually start my timer and keep an eye on things. I wanted to speak today about really how it is that we see changes coming in medical research, specifically with regards engineering and computational challenges that are coming our way, and some of the reasons why it is that we've established here at Garvan a data sciences platform. So I thought I'd begin with a bit of an unusual sort of way and ask you to imagine the following experiment. We've got two mice. One is rather chunky and the other one is quite lean. One of these mice was fed lettuce. The other mouse, he actually preferred cheeseburgers, chips, and Coke. Now, the interesting thing is that many of you might have thought that it was this mouse over here that was eating this diet. But in fact, it's the other way around. And we're particularly interested to understand what is it about this mouse over here that allows him or her to eat this kind of diet and yet not put on weight. So we might begin typically by looking to be able to measure things about the mouse. We would go and weigh it. We would typically weigh the chunky mice, we would weigh replicates of the lean mice, and we would look as to whether there was in fact a statistical difference between the heavier ones and the more lean ones. We might also say, gosh, well, on this type of diet, what's the level of salt in the blood? So we might look to understand what's the potassium ions or the sodium ions inside the animal. In addition to that, we might also say perhaps we should measure the cholesterol levels, given the fact that these mice are on such very, very different diets. And the key point I'm wanting to make is that with all of these types of experiments, we would really only be measuring single data points. We might say that the heavier mouse was 55 grams compared to, say, a 20-gram mouse. We might say that the sodium concentration was this much compared to that, or that the cholesterol was this. Each time it would just be a single data point that we're measuring. Now, Mandy just gave a beautiful overview of DNA, and I might just want to emphasize a few points. When we talk about DNA, and we're talking really about these letters A, C's, T's, and G's that then go on to pair up with others and form a beautiful helical structure that eventually gets packed up into chromosomes, and all of the chromosomes together constitutes all of our DNA makeup or our genomes, that in fact what we're looking at over here are a total of around 6.4 billion letters. So that's 6,400 million letters, or about 3.2 billion paired letters that we're talking about. And each one of these letters now forms a data point. So in contrast to measuring the sodium or measuring the mass of the mouse or measuring the cholesterol of a mouse, whenever we're now sequencing a genome, and typically the genome of a human being, we literally get out of that study 6.4 billion individual letters that can now become informative to us. So the first human genome was sequenced way back now in 2001, when in February, on the cover of the journal Nature, the human genome was presented to the world. And an awful lot has happened in the interim in terms of the kinds of technologies that have evolved in order to be able to sequence genomes. But that sort of evolution of technologies is far clearer when we look at this graph. What we're looking at over here on the vertical or the y-axis is data on a log scale, and this represents the cost of sequencing a genome back in 2001, and this is time from 2001 all the way through to the modern day. So at the time that the human genome data was presented to the world in that Nature Journal article, it cost $100 million to sequence one human genome. In contrast, today, it costs approximately $1,000 to sequence a genome. And the implications of that are multiple, but one that I would particularly want to highlight is back in 2001, when a human genome cost $100 million, the entire world shared one human genome. Today, in contrast, where a human genome costs around $1,000, we literally at Garvan have PhD students and groups, such as Mandy's group, that would be looking at hundreds and even thousands of individual genomes. So it's quite extraordinary that if we were to look at the value of a genome back then and say, Garvan, as a research institute to date, has sequenced approximately 25,000 human genomes, that would represent a value in 2001 terms of approximately $2.5 trillion. Now, these are just extraordinary amounts when we think of how it is that this extraordinary investment in technologies has actually brought the cost of genome sequencing down so dramatically. So, in order to define to you or to describe to you why it is that we're needing to establish a data sciences platform at Garvan, I'd like to represent this graph. So here what we're talking about on the vertical axis is data size, and the data is going to be on a log scale. So every time we're looking at a doubling of the data, it's actually a 10-fold increase in the size of the data. And again, on the horizontal axis, we're looking at time. What I'm talking about over here would typically be typical Garvan deliverables. Those would be impactful science, patient treatments, or how it is that we can positively impact on the health of patients, examples such as what Mandy just alluded to, and then, of course, opportunities for commercialization through patents and such like. So, when we were initially starting to sequence DNA, we were just looking at very, very small parts of the DNA. And what we saw at that time was the data sizes were incredibly small, and the proximity of the data to science, patient treatments, and patents was also very, very close. But as these technologies evolved, what we saw was that the size of the data began to increase, and there also started to appear a gap between the data and our ability to translate that data from this layer all the way through to where it is that we can begin to deliver on our scientific insights. As technologies grew even further, what we saw was a dramatic increase in the data itself, and then associated that was an even further increase in the distance between the data itself and how it is that we could achieve that translation. And then, as Mandy alluded to, at Garvan we run effectively a factory-scale genome sequencing operation. The data has become much, much larger, but so too has the distance away from this red line at the top. So the way in which we see things is that at this sort of level, bioinformatics or data sciences were able to start providing that or bridge that gap between the data and the red line and getting data across or getting the science across. As the data sets started to increase further in size, and here we're already talking about petabyte sizes of data, so those are thousands of terabytes of data, we now needed to introduce an engineering layer to the data itself. And then in addition to that, we needed to actually add an analysis platform. So it's at this sort of level over here that our new team that we've created at Garvan, the Data Sciences Platform, operates. It's there to be able to bridge that gap between the very, very large data sets that we're generating today and being able to put that into the hands not only of bioinformatics community here at Garvan, but also research scientists to be able to get to start delivering on our science itself. Okay, so the key areas in which we as a data sciences platform are now working are in the areas of what we call production data processing. That is how it is that we could take raw genomic sequencing data and convert it into a point where we can begin to understand what is the genetic variation that we're seeing inside every single patient genome, how it is that we can support our user community through user interfaces, the underlying computational infrastructure that we've built not only at Garvan, but how it is that we partner with supercomputing facilities in Australia, specifically the National Computational Infrastructure in Canberra. And then in addition to that, the sizes of the data sets that we're generating now are so very, very large that it's no longer really feasible to move the data, and instead what we look to do is to bring the computation to the data so that we can analyze the data in situ rather than actually doing things the other way around. I'm just mindful of time, and I still have 5 minutes, but I don't want to lose out on too much of the opportunities. So if I were to go back, Garvan as a research institute, we work across 3 themes: cancer, healthy aging, and then inflammatory diseases as well. And the way we see things evolving is that genomics underpins so much of what we're doing. Genomics is not a one-stop shop to all of the problems, but it does provide for us this complete layer of information upon which we can build so much more. But it is literally within that point of that complete layer of information upon which we're trying to build that we now have to be cognizant of. We have those 3.2 billion paired data points that we're looking at in every single genome. Every time we're making some sort of a scientific discovery, that might depend on only a single data point. In some cases, in the areas of typically polygenic diseases, we might be looking at multiple data points. But it's vital for us to be able to be keeping all of that data so that we can interrogate parts of genomes and parts of patient genomic information at any given time in the future. And this will assist us in our evolution of becoming a truly data-informed medical research institute. In addition to that, what we've also seen is that the type of science that we do at Garvan is changing. Historically, we might have begun with studying individual cells inside Petri dishes. We might have looked at individual mice or animal models in order to study disease. And then finally, the idea was the translation into person. So the direction of travel was very much in this way, from the Petri dish across to the human. With the coming of our ability to be able to sequence human genomes at scale, we're actually able to reverse this process by studying humans first and then looking to validate our findings inside mice and then also in cell lines as well. Okay, so I'm going to fly now because I've got too many slides and I'm running out of time. But some of the areas in which we as Garvan have been working is to build patient cohorts. Back in 2014, the New South Wales government funded what's called the Sydney Genomics Collaborative, working in 3 main programs: the establishment of what we call a medical genome reference bank, individual research grants that were awarded to researchers across New South Wales, and then also a cancer medicine program. The research grants that were awarded across very, very diverse disease areas, and we're fortunate to have sequenced the genomes of these individuals and now looking to be able to bring all of that data into the hands of both researchers who have been approved access to be able to use that sort of information, as well as research active clinicians. We're also working nationally in what's called the Australian Genomics Health Alliance, and this is a rather busy slide, but we're supporting the housing of multiple flagship patient cohorts in very, very diverse disease areas. In addition, more recently, the federal government of Australia announced what's called the Genomics Health Futures Mission, and this is a $500 million initiative that will run over the next 10 years. And the initial phase of this is the so-called Mackenzie's mission that looks to identify potential carriers of genetic disease. And again, our group will be supporting this initial phase of what's called the McKinsey's mission. We're also fortunate to collaborate with what's called Circa, and this is primarily looking in the areas of individuals who are born with innate problems in their immune systems. They have defects in the DNA of their immune systems, and delighted to be able to participate with that as well. So our overall workflow begins with individual patient groups. From there, we look to be able to provide the underlying computing resources. As I mentioned, we compute at Garvan, we compute at the National Supercomputing Facility, and we also compute on commercial clouds. We run these production workflows in order to take raw sequencing data to the point of variance. We're looking to be able to share that data responsibly so that only approved users actually have access to the data. We're wanting to be able to support the administration of the sharing of this data. And finally, we've also built what we would call the Google for genomics today, what we refer to as our Vectis platform. Vectis is called a lever, and it's there to be able to support those kind of analyses. I see I'm out of time, and so I'm actually going to stop here and maybe just progress to the ends. And sorry, I had too many slides, but the last part I just wanted to mention is that Garvan today, the demographic of Garvan comprises bench scientists, it comprises research active clinicians, and what we're also seeing though is the changing of the demographic of the coming of engineers, both computing engineers, software engineers, data scientists that are contributing to the change of this most wonderful environment in which we work. So I'm going to stop there now.